Everyone, welcome. Good afternoon, good evening, good morning, depending upon what time zone you're in. My name is Greg Fine, and I am board chair of Great Basin National Park Foundation, and you are taking part in the third installment of our 40th anniversary speaker series presentation. And the foundation is the organizer and producer of this series, and it's all part of our gearing up for the 40th anniversary of Great Basin National Park when the park became an official park in 1986. So, just a little bit about Great Basin National Park Foundation. We are the official Friends organization, and we help the park support the park through a variety of programs and funding avenues. And we are a pretty small but mighty committed group to the park's success and making sure that it is protected and available for generations to come. If you would like to learn more about Great Basin, visit greatbasinfoundation.org and you can sign up for the newsletter and what have you. Before I jump in and introduce Dr. Ali Coconis, a couple of housekeeping items I'd like to just clarify. Uh, Dr. Coconis is going to talk about pack rats, wood rats for about 45 minutes, and then she will entertain questions for the last part of the presentation. And you are free to leave your questions in either the chat or the Q&A section on your Zoom window there. She won't be taking questions during the presentation, so just know that Claire will facilitate getting to your questions at the end. Um, Now let me talk to you about this program. So this program on pack rats or wood rats, the name is interchangeable, will explore both the ancient and contemporary significance of pack rats, highlighting the species' ecological roles, the scientific breakthroughs made possible through their midden analysis, which is their nests, and the ongoing questions that continue to drive the research forward. Leading the charge on that and spearheading our efforts tonight is Dr. Ali Coconis. She is curator of mammals at Santa Barbara Museum of Natural History, where she oversees more than 10,000 terrestrial and marine specimens and supports their long-term care and research use. She also conducts her own research on small mammals and is currently preparing work for her doctoral studies for publication, while also exploring fire ecology in Southern California. Dr. Coconis brings more than a decade of museum experience, including roles with the History Museum of Utah in community outreach and collections, and the University of Nevada, Reno Museum of Natural History as graduate student curatorial assistant. Say that three times fast. Tonight, she will present on the history and importance of small mammal research in Great Basin, with a particular focus on pack rats and their middens. Her talk is titled Packrat Middens, Uncovering Thousands of Years of Biotic Change in the Great Basin. And with that said, Dr. Coconis, I turn the spotlight on you.
Wonderful. Thank you so much, Greg. And thank you to Claire and Aviva at the Great Basin National Park Foundation for inviting me today. And though I don't live in the Great Basin anymore, it's been a big part of my formative years in my professional and personal life, and will certainly continue to shape both. And so with that, I also want to thank the foundation in helping to support Great Basin National Park, particularly in a time of much needed funding. In that same vein, I'd also like to start with showing my appreciation for museums. Many of the opportunities I've had to explore the natural world and learn about science has been through my connection to museums and to the wonderful humans that work in them. I'm going to give a special shout out to the Natural History Museum of Utah, the University of Nevada, Reno Museum of Natural History, and now to the Santa Barbara Museum of Natural History. These are organizations that truly inspire and support ecological research, and I'm very thankful to have found a career in museum science. Oops. Is it going to let me advance? Yeah. Okay. So Western North America's landscape is quite mountainous. And right in the center of this mountainous terrain is the Great Basin Desert. And this region sits sandwiched between the Rocky Mountains to the east and sits in the rain shadow of the Sierra Nevada on the west, which produces a cold temperate desert. And it has surprisingly, over 300 named north-south trending mountain ranges with these intervening valleys, creating these local habitats of desert shrub all the way up to alpine tundra, which you can see here in this photo. And the mammalian species diversity in this region really reflects this habitat diversity. So, Badgley et al. Shows here in a series of papers that across the longitude of North America, so from the Atlantic Ocean on the east all the way to the Pacific, there's a lot of variation in the topographic relief or the change in the elevation over area, and that it significantly increases from the Great Plains into the Rockies and then over the Great Basin to the Pacific Ocean. So basically just showing what I just said, which is that it's very mountainous. And there is this really strong correlation between this mountain terrain and the mammalian species diversity, with high species diversity out west, as a similar profile as you can see to the topographic relief. And thus, this has really sparked an interest in this region for quite some time. And certainly a major focus of research throughout this region has been on small mammals. And this is because not only are they diverse and relatively abundant, making them easier to study patterns and processes, but they're quite sensitive to environmental change. They have these relatively small home ranges, unlike larger mammals or birds, for example. And wood rats in particular, estimates of daily foraging distance are as small as 100 meters. And so this makes it really difficult for them to leave if there are major disturbances. And they often have very specific habitat requirements. So for example, kangaroo rats, pictured here in the middle, prefer particularly sandy soils in which to build their burrows, or the American pika, they're generally confined to these high mountain talus habitat because of their extreme thermal sensitivities. And furthermore, they have relatively short lifespans and produce a lot of offspring typically. So this provides pretty immediate feedback for environmental change. So if there's a disturbance, usually the changes in abundance and diversity occur in these animals quite rapidly. And many people over the last century have had a fascination with small mammals in the Great Basin for some of the reasons that I just mentioned. So really starting in the early 20th century, land use change was sort of rapidly occurring across the American West, mainly in the form of mining and agriculture. And a prominent biologist pictured here by the name of Joseph Grinnell recognized these significant changes happening. And he decided to document this biodiversity before it was gone or at least significantly altered. So in the first few decades of the 20th century, he and a team of researchers set out across California and Nevada to set these baseline records of this region. Here he says, after the lapse of many years, possibly a century, the student of the future will have access to the original record of faunal conditions in California. At the time, he was the director of the Museum of Vertebrate Zoology at UC Berkeley, and so California was the initial focus, but many of the mountain ranges in Nevada were also surveyed, most prominently by one of his students, E. Raymond Hall, who later writes the Mammals of Nevada book. But maybe more interestingly, a woman by the name of Annie Alexander was interested in and funded this whole endeavor. She had a really interesting life. She was a Hawaiian sugarcane heiress, but had an interest in the natural world and played a heavy hand in the formation of the Museum of Vertebrate Zoology at Berkeley. And she had a particular fascination in paleontology, actually. On the left, this is a photo of her in a dig in the middle of Nevada in the early 20th century, unearthing some ichthyosaur fossil specimens that some of the best specimens that we still have today. And on the right, this is her pictured. She's on the left, and then she's pictured with a colleague of hers, Louise Kellogg, who together they would actually go out into these remote places all over the West, including some trips up into like really rural Alaska, and conduct these faunal surveys, which was really quite unprecedented at the time, and I find quite inspiring now. During these early surveys, photographs were taken of some of these places, which documented the general vegetation across these mountainsides and throughout these valleys. So the MVZ, just to note, the MVZ, the Museum of Vertebrate Zoology at Berkeley, has many of these images online if you're interested. There's a lot more than this. But these are two examples from central Nevada, one in the Toyabe Range, which is right here in the center, and one in the Ruby Mountains up in the sort of northeast corner of Nevada. These historic photographs have since been compared to more recent photographs showing pretty significant vegetative change over time. In particular, the Great Basin has seen an expansion of the pinyon juniper forest, or what most of us colloquially call PJ. And this is because across the Great Basin, these two species, pinyon pine and juniper, have a very strong association with one another. So you might hear this throughout the talk, me referring to PJ. And they make up a pretty large portion of the vegetation across this region. But these expansions are occurring and taking over what was once mostly sagebrush habitat. And this expansion has been attributed to a combination of factors, including things like land use change, such as shifts in agricultural practices and fire suppression, as well as climate shifts. And of course, these major vegetation shifts can have these downstream consequences for resource use and distribution of native fauna. And some interesting, significant work that's utilized these baseline surveys or studies that evaluate changes in the faunal elevation ranges over the last century. And there have been several of these conducted, and one most notably in the Sierra Nevada across Yosemite National Park. And it was published in the Science paper back in 2008. But also a study of this sort was completed in the Great Basin. And so this is part of it. So in general, the Great Basin has experienced this warmer and wetter conditions over the last century, quite surprisingly. And though there's certainly a lot of variation across this very large region, this trend is quite dominant and expected into the future. And in a study done by ecologist Dr. Rebecca Rowe and colleagues, they assessed elevational shifts over an 80-year period in small mammals in the Ruby Mountains, again highlighted down in the bottom right, showing that for the most part, these species range limits are not stagnant, and that they certainly move in ways that can be quite variable. In accordance with some predictions, though, they found downslope range expansion by a PJ specialist, the pinyon mouse. And as well, decreased occurrence in at low and mid elevations by sagebrush-loving species such as the Great Basin pocket mouse, the least chipmunk, and the sagebrush vole. And this, of course, reflects these vegetative shifts occurring, namely the expansion of PJ in this region, which seems to be benefiting from the warmer and wetter conditions. Okay, packrats. As Greg mentioned, they're also called wood rats. These are synonymous names, so I'll probably stick with wood rats throughout this talk, but I also mean packrats. These animals, on the other hand, can be quite resilient, but over the long term have also revealed these similar elevational shifts as climate has changed. So wood rats are native North American rodents. And so just to clarify that despite being called rats, they're not as closely related as you might think to the non-native black or brown rats that you would, for example, find in a city. In fact, they are in two completely different families. So they're pretty far apart. But what makes them special is their high species diversity across Western North America with about 22 different species, making them a really great system to study patterns and processes of speciation, or the ways in which speciation happens and how diversity is maintained. And so this is just here on the right is just a panel from the Peterson Field Guide to the Mammals of North America. It's drawn by Fiona Reed, and I love it. It's only showing 8 of the 22 species, but I think it really displays some of the color and size variation that you see amongst this genus. But across the Great Basin, there are 2 species that are known throughout, and the 2 that were the focus of my PhD work: the bushy-tailed wood rat, or Neotoma cinerea, which is shown in the blue arrow and pictured on the bottom left there, and the desert wood rat, or Neotoma lepida, which is shown with that orange arrow and pictured on sort of the center of this photograph on the bottom. And these colors these blue and orange colors will be consistent throughout the talk for these species. The bushy-tailed wood rat is larger and has thicker fur and is mesic adapted, meaning that it typically lives in colder, wetter environments in comparison to the desert wood rat, which is smaller and has a thinner coat adapted for hotter, drier environments. And these 2 will be featured more prominently a little bit later. But now I'm going to talk about their nests. In general, all wood rats have really interesting nesting behavior. They are very busy nocturnal animals. Throughout the night, they are continuously running in and out of their nest and building and modifying these structures. In wooded regions, like in the California oak woodlands, they typically build these nests at the base of a tree or a shrub, as pictured on the top, the 2 pictures on the top left. But in the Great Basin, their nests are pretty confined to rock. They either find a boulder that they can crawl under, or a crack in a rock that they can crawl into. And these rocks kind of form the skeleton of the nest that they pile various plant material on. And excavated nests have shown that it's quite complicated inside. So these are some diagrams from Stones and Hayward from 1968, where they excavated a couple of nests. And it shows that they have, lots of different chambers for things like caching food and rearing offspring, and many tunnels and entrances and exits for escape routes if necessary. And perhaps more notably, these animals display some pretty funny behaviors of collecting and hoarding objects, hence the name Packrat. And so if you look closely at a wood rat nest, you'll find lots of things like small bones and piles of rock that they've sort of made into a mosaic. And even sometimes bottle caps and pieces of glass that humans have left behind. In fact, I've always wanted to do this on a wood rat talk. I'm going to read you a small passage from a really great book called The Mammals of North America. It's by Victor Kaelin, and it was published in 1947. And I think it really speaks to the sort of whimsy of these animals. In the Western mountains, one sometimes hears of lifelong partnerships being ruptured by mysterious thefts. The loss of money or a watch may sow the seeds of suspicion, only to have the missing valuables turn up later in a nearby wood rat's cache. Some Colorado miners once missed several pounds of tallow candles. Later, one of the men found them carefully piled on a ledge behind the timbering, about 35 feet down the mine shaft, where a wood rat had taken them. Even dynamite sticks have been lugged off and hidden. Prospectors who have carelessly left their false teeth beside their bunks at night have lisped and gone hungry on a soft diet for days afterward. I've had the shoelaces stolen out of my shoes. One Allegheny rat went so far as to go off with things it didn't want. Finding a woodsman asleep on a bed of small hemlock boughs, it could not resist its obsession for thievery. Stealthily, it pulled every bough from beneath the man without waking him and scattered them about the nearby woods. So they're quite characters. But of course, these nests are not just to hoard their loot, but also function very effectively as thermoregulators. And this study was done in Death Valley National Park at the valley bottom, where temperatures in the summer can exceed 120 degrees Fahrenheit, so really hot. And amongst these large honey mesquite bushes that you see pictured here, desert wood rats build nests. And in this study, they attached temperature loggers to the back of these desert wood rats, insulating them from their bodies to measure the ambient temperature of their nests in comparison with the outside temperature and detect when they were foraging at night. So these data and this plot here come from one individual wood rat with time on the x-axis here, 24-hour period, and temperature in degrees C on the y-axis. And this red horizontal line here is the upper lethal temperature of these animals, which is around about 42 degrees Celsius or 107 Fahrenheit. And the three red lines here show ambient temperature outside of the nest at varying height above ground level, and the black line is the wood rat. And so you can see that throughout the day here, the wood rat is in its nest and really only emerges in the evening when the ambient temperature outside falls below that upper thermal limit or upper lethal temperature. And also that nest was quite insulating, sometimes up to 15 degrees Celsius or 20 degrees Fahrenheit as a buffer. In addition to the thermal regulation of these nests, they provide a lot of protection to the desert community. In fact, these nests have also, we also find that they have a higher relative humidity, as much as 28% more humid than the outside, and provide an added layer of protection from desiccation for animals, which is important in these super dry places. And beneath these nests, the soil is more nutrient rich from all the organic material that they gather, as well as their own feces that are on or around their nests. And so these nests attract a lot of other animals, creating what has been dubbed centers of biodiversity. Arthropod diversity increases in these nests, including crickets and wolf spiders and various beetles, as well as other vertebrates from lizards to mice, even gopher snakes. In fact, desert wood rats actually live sometimes in and around rattlesnakes, though the exact proximity between those two is not fully known. But still really fascinating. And just as a side note, to talk about really how cool these wood rats are, desert wood rats are preyed upon by rattlesnakes. No surprise there. And this is across the range. But in response, they have developed a resistance to rattlesnake venom and can actually survive if bitten. But this is very dependent upon other stressors in their environment. So recent work has shown that temperature and diet can actually affect the ability for them to metabolize this venom. For example, in colder environments and with a diet that's high in creosote, which is a native bush that sometimes grows in abundance, these combined stressors might overwhelm their system and decrease their ability to metabolize the snake venom. But in addition to the modern research on their nests, there's been quite a bit of paleontological work involving wood rat nests. And I previously mentioned, wood rats, build these extensive nests where these debris piles of organic material, including to a large degree plants, but also bones and feces, including their own, are then encased in their own urine. So they pee all over it, and this hardens and solidifies into a big mass, and this is typically what we refer to as a midden. And over many generations, many generations, these pilings can build up, like this photograph on the right here, and form what we call a midden series. And this can capture actually tens of thousands of years of biotic change at a single locality. And these are just some examples of middens inside a cave in Great Basin National Park where layers of organic material and wood rat urine has glazed over and turned into what we call amber rat. And this is, of course, in reference to amber or hardened tree resin that has preserved fossils. But in this case, with amber rat, it's feces and urine, preserving mostly what we call subfossils or ancient remains that haven't fully fossilized. And within these middens, there are, of course, animal and plant remains, indicating shifts in these groups over tens of thousands of years. But even they also contain archaeological specimens, so traces of human existence at these cave or shelter sites, which is also really cool. And this was a major focus of wood rat research for quite some time in this region. Over the past 50 years or more, researchers have excavated now over, this number is a little old, so it's now over 2,000 middens, which are mostly stored at a repository at the University of Arizona. And these middens vary in age based on radiocarbon dating, providing information at both the broad and at the fine spatial and temporal scales. At the broad scale, major climate oscillations have occurred in this region, leading to shifts in the hydrologic landscape and modifications to plant and animal assemblies. So during the last glacial max, which was about 18,000 years ago or so, large glacial lakes were widespread across this region, including two popular famous lakes, Lake Lahontan and Lake Bonneville. And they have been, though, since greatly reduced, opening up habitat and corridors for dispersal for dispersal of terrestrial species across this region. And many of these lakes are completely dried up. And you can really see that in the wood rat midden record. So this graph here shows the number of middens found in the Great Basin over time. But before the last glacial max, here, let me get my arrow. Before the last glacial max here, there weren't a whole lot of records, as you can see. But following this period where you see glacial lake retreat, we have this uptick in the number of records that we've found, a number of wood rats. But then this region hit a very hot and dry period known as the mid-Holocene between 8 and 5,000 years ago, and the midden record declined once again. And so this region-wide record really closely follows what we know of the climate cycles in this region. And at a finer spatial scale, these middens reveal plant community change through time that also coincides with these major climate shifts. So for example, a single midden from eastern Nevada at around 2,000 meters or 6,500 feet shows this shift from the more boreal species, so bristlecone pine and Engelmann spruce. These are species that we see at much higher elevations than 2,000 meters today, to a species that we see at 2,000 meters today, or this PJ woodlands, the pinyon pine and Utah juniper, and that this shift occurred around the early to mid Holocene, which is again that very warm and dry period. In addition to this fine scale information about their environment, we also see wood rat species identification shifting over time. And it turns out that their fecal pellets, which you see me holding here, they're highly correlated with body size in these rodents. This graph here shows that very positive relationship. The larger the mass of the wood rat on the x-axis here, the larger the fecal pellet on the y-axis. Sorry, I'm getting notifications that I need to turn off. Okay. And so with this known relationship, researchers were able to measure change in the body size over a much longer period of time in wood rat middens. Oops. No, it's not letting me switch. There we go. So here they're looking at multiple middens across an elevational gradient in Death Valley once again, showing a precipitous decline in body mass in the bushy-tailed wood rat since the last glacial max. And we know it was bushy-tailed because of the size. It's the modern bushy-tailed and desert wood rats have there's sort of a gap between the size of the two, and so the desert wood rat wouldn't have been this big. So we see the decline though in the bushy-tailed wood rat body size since the last glacial maps. This is time over thousands of years. And then it sort of levels out, but then we see the emergence of the desert wood rat into this record. And again, this is over an elevational gradient. These are multiple nests. And so though they are both in existence for a long period of time, they're elevationally separated. And then eventually a complete species turnover happens where we actually have no more records of the bushy-tailed, and this is what we see there today in Titus Canyon in Death Valley National Park. Only the desert wood rat remains. And this relationship and elevational pattern continues to hold up through time and space in this region where the mountain topography has allowed these species to move upslope as climate has warmed and downslope as climate has cooled. But for the most part, their populations remain totally distinctly separated either across mountain ranges or where both species exist in a single mountain range by elevation. Whereas expected, the larger bodied species, the bushy-tailed wood rat, is at higher elevations, and the smaller bodied wood rat is at lower elevations. And these are here, these three are representations of large elevational gradients in Nevada, and these are modern records that show this pretty distinct elevational distribution from the Wasatch Range in the western Nevada to the Toyabe Range in central Nevada, and then the Snake Range in eastern Nevada. And though not perfect, very distinctly what we would expect. And so my interest in the system really revolved around this distribution and the mechanisms by which they maintain this distribution through time. And despite what we saw in the modern record, we predicted there to be these sort of narrow bands of co-occurrence at mid-elevations throughout this region and in very select mountain ranges. And finding these limited localities would be really key to probing for these mechanisms by which they maintain this distinct distribution and maintain this diversity over time. And so first I had to find these localities. This was somewhat like finding a needle in a haystack because at the time, there were really few to no records in the search databases that had indicated any major co-occurrence across this region. So the first step for me was modeling their distributions, and these models are based on modern records and climate. I built models for both species, and then I overlaid them to find where they could potentially both co-occur. And the predictions for co-occurrence are mostly at these sort of mid-elevational bands across these mountain ranges, and you can really see that here in these mountain ranges here in Utah and the Snake Range there. So the purple here indicates this co-occurrence. And then in step 2 was to go into the field and ground truth these models. And so I did this in the Southern Snake Range or in Great Basin National Park, which indicated by the models that there are localities where there's this high potential for co-occurrence. And lo and behold, the models worked and quite well. We found many sites, I've highlighted 4 here, where these two species were living together, co-occurring across a mountainside. This, again, they generally coincided with our models. So one of the major findings was not just this co-occurrence, but that both species were sort of very, very closely following climate across this mountain range because our models were built upon climate. And so this was an indicator, this is an indicator of the likely effects of future climate change. And I just wanted to give you a better visual of one of these sites of co-occurrence. And so here I've plotted it. So each dot here represents a single individual that was caught, and they're colored again by species. So orange for the desert wood rat and blue for the bushy-tailed wood rat. And you can see here that we caught both species well within their home ranges of one another, so the potential for interaction is quite high. Okay, before I proceed though, I'm really going to make a quick plug for Great Basin National Park because it's a really special place to me. This is where I did the majority of my field work during graduate school, and I feel very blessed to have spent so much time in this really gorgeous place. It's located in very far eastern Nevada on Route 50, really on the way to nowhere, but it's definitely a destination in itself, and it has as I've been talking about, it has this really incredible elevational relief with over 8,000 feet of elevational change from the valley bottom to the top of Whaler Peak. You get these expansive views. It's incredible. And it's a dark sky park with really amazing astronomy programming. It has remnant populations of bristlecone pine forest. Here I'm standing with one that has been dead for thousands of years. These trees are so dense that they will, even after dying, they will still stand for thousands of years, which is really crazy. And it has these extensive caves throughout, and they're known for these shields here in the cave. So really amazing. This is Lehman Caves, so one of the caves that you can tour. But to collect data on wood rats, so we would live trap them using these traps here, and we would open the traps, so they're nocturnal, so we would open the traps at dusk and then check traps anywhere from midnight to 4 a.m. So we were quite nocturnal as a result. And then for each individual, we would take a bunch of data. We would take body measurements. We'd take a small clip of their ear, just really, really tiny little clip for genetic data, and a sample of fresh poop for diet and microbiome. We knew it was fresh because we would sweep under the trap every night to make sure we were getting these fresh samples, and then we would ear tag each individual so we could identify recapture. And the fecal pellets were then placed into cryogenic tubes and immediately flash frozen, meaning we would store it in liquid nitrogen in the field, and this was mainly to preserve microbial composition. And then in addition, we collected a few whole specimens that represent both species that are now accessioned into the UNR Museum of Natural History. And over the course of 3 years, we had a total of about 500 live captures consisting of about 280 unique individuals, which was a huge effort, and I'm very thankful for all of the people who helped on this project. And so I'm going to go into some of the results of my graduate work, but this certainly won't be comprehensive, so I'm happy to chat more after if people are interested. And so an interest of mine was in habitat selection, and one of the factors I wanted to measure was nest microclimate. So really kind of in an attempt to emulate the study in Death Valley that I previously mentioned, I backpacked wood rats with temperature loggers. But attaching backpacks to live squirmy, bitey wood rats is actually a lot harder than it may look, or maybe it looks really hard. I don't know. It was difficult. They're absolute magicians in getting out of these things, so that was quite a challenge. And so my incredibly patient field techs and I got to the point of developing these great backpack designs, but we didn't have enough time to gather all of the data. But because we designed these backpacks, I'm really interested in using this technique in the future on wood rats and other species to look at microclimate. However, we did use a different method to measure various nests in this site of co-occurrence that I showed you earlier, which produced some interesting results. So I was interested in the difference between these nest temperatures to show niche partitioning between these species. And this is the setup here. So there was a nest under this rock, so we caught a wood rat there. And for the external measurement, I placed a temperature logger on a wooden stake about a half-meter off the ground with an aluminum pan on the top to block any sort of solar radiation from getting to the temperature logger. And then I placed another logger on about a 2-meter long fiberglass rod that was then kind of fished up inside of the nest as far as possible and then secured with rocks because they would certainly try to move it. And I measured both the internal and external nest temperature of 5 bushy-tailed wood rats and 4 desert wood rats for an entire year and taking a measurement every 30 minutes. So lots and lots of data. And these are the results. For these graphs, both the summer months are shown here on the left, June through August, and then the winter months, December through February on the right. And so I've only taken a portion of the data that we collected, and these are just like the extremes of the temperature data. And so for these graphs, we have, again, time of day on the x-axis in a 24-hour period, and then nest temperature buffer on the y-axis in degrees Celsius. And so this is the average differences between the external and internal temperatures. And so what this is showing you is that really anytime that these lines are above the zero mark here or here in the four graphs, it means that the temperature of the nest was warmer than the outside air temperature. And anytime it was below these lines, it means the temperature of the nest was cooler than the outside air temperature. And the results really show some interesting things. All but one of the nests, and of course these are colored again by species. The bushy-tailed or the larger wood rat in blue and the desert wood rat in orange. All or I should say 8 of the 9 nests were showing that they were quite insulated. So during the nighttime hours, the nest was warmer than the outside air temperature, and during the daytime hours, it was cooler. And this is pretty much the case for both the summer and winter seasons. So that was kind of interesting. However, at one site shown here by this asterisk, it was occupied by a bushy-tailed wood rat. It was particularly insulating, meaning that for all hours during the summer months, it was actually cooler than the outside air temperature, including at nighttime. And then in the winter, for all hours, it was warmer than outside air temperature. And this is significant because this nest was kind of a throne of wood rat nests at this St. Patrick's site or the site of co-occurrence. It was built in this crack system. This nest was built in this crack system in the back, way back of this protected shelter that you see here. And what makes it so interesting is that these sites, these really protected sites, are quite limited on the landscape. And they would certainly be favored by both species for their protective thermal qualities. So they're really kind of likely an important source of competition between these species, which makes sense that the larger bushy-tailed wood rat was found here rather than the desert wood rat. And these particular sites may also be really good sources of climate refugia for these populations, allowing for really longer term persistence of wood rats at these sites. So think about 100 years or 200 years. These species can remain here because of these really small microrefugia sites. Okay, and so next, I was interested in the diet and microbiome between these species, which is why I collected all of that poop. So wood rats in general are herbivores, consuming mostly foliage and fruits, and are well known for their facultative dietary specialization, meaning that across their large ranges, so the desert on the left and the bushy-tailed on the right, they have really large total ranges. They consume a lot of different plants throughout these total ranges. So they're considered dietary generalists. But locally, they can and do consume a more specialized diet depending on what is available. In addition, these plants can be quite toxic and often are. But through specialized gut microbiome and genetic adaptations, wood rats are better able to consume these plants than other animals. In the Great Basin, for example, both species eat juniper, which is high in terpenes and phenolics, which are plant secondary compounds, and they aid the plant and the prevention of herbivory typically, unless you're a wood rat. And here are the results of the diet analysis, which very much indicates the sort of species and site-specific differences in these between these two wood rats in diet. Most notable are differences in juniper consumption between species, as shown in the green coloration here and in these pie charts. The pie charts here at the bottom represent the percentage of the top plants, shown here on the right, consumed across this range by both species. So not both species don't consume all of them, but they are the top plants at each of the sites we measured. And at these sites of co-occurrence here boxed in the center, site 1 and 2, the desert wood rat we see proportionally consumes more juniper than the bushy-tailed wood rat. And this was actually predicted as the desert wood rat may more efficiently metabolize, clear some of these plant secondary compounds from toxins in the juniper than the bushy-tailed. And in some populations of desert wood rats, their diet has been found to be to make up greater than 90% of it is consistent of juniper. So they can really consume a lot of this plant when needed. And so though both species, but to a greater degree, the desert wood rat, they are facultative specialists when exposed to a large amount of juniper on the landscape. Sorry, I kind of lost my mouse. And they, so they're special. They can specialize on juniper when needed, and they eat less so, kind of at higher and lower elevations, as indicated by these pie charts. And this is sort of generally due to lower abundance of the plant of juniper at these sites. And these results certainly have implications for the projected growth of this forest type across this landscape, as well as the ability for these wood rats to cope with changes over time here. And finally, we examined the gut microbiome of these individuals. As I previously mentioned, microbes in the gut have been found to aid in plant digestion, as well as perform other regulatory metabolic functions in these animals. And because wood rats are known to consume highly toxic diets, they've been studied quite extensively. And so these were the results. So this is a bar chart showing relative gut microbial abundance in the bushy-tailed wood rats in the sites, these blue sites here, and then in the desert wood rat and these across these orange sites here. And so the bacterial families that we found within their gut are colored and shown again in the relative abundance for each of these bars. And though it's not super easy to see from this chart, we actually did find a really strong species-specific signal of gut microbial composition. And so what this means is that, like most mammals, their gut microbiome is inherited mostly from their mother. So it's passed down, though they can still obtain some of it from their diet. Another really interesting result was that we see really familiar patterns in the presence of particular toxin known to aid in plant digestion. Sorry, I keep losing my mouse. Okay. So the 8 bacterial families that I've listed out here in those colors, they're made up, so they make up about 97% of the total sort of relative abundance in all 97 of the individuals that we tested. And they're composed of only 4 phyla. So this is a higher taxonomic group, 3 of which, Firmicutes and Bacteroidetes and Actinobacteria, are really known to dominate the gut microbiome of communities of herbivorous mammals. And they perform interesting functions, like allowing for the metabolism of starch and cellulose and xylem. And these might be particularly important for plants in the Great Basin, like the juniper or others that are highly fibrous. In addition, certain members of firmicutes are also known to aid in the detoxification of plant secondary compounds. In particular, Lactobacillus is a genus in the family Lactobacilliaceae. This family was the most abundant microbial family in both species in our study. Lactobacillus likely aids in the degradation of phenolics in juniper. So it's very interesting correlations that coincide with what we know of the metabolic functions performed by some of these microbial families here. But with all of this, I really just want to drill home the real strength of this wood rat system, which is the ability to integrate both the contemporary results from the study into what we know of their paleo distributions, as well as some molecular work that's been done, a whole field that I didn't even go into today, including some really incredible work with ancient DNA. And altogether, the pieces of this puzzle provide this sort of more complete picture of the patterns and the processes across both space and time in this region. And that is what makes these animals really interesting, this system really interesting. And so that is what I have to share with you today. But I did want to say one last thing before I end this talk. I was asked to share sort of a brief overview of my background. And though Greg went over this in the beginning a little bit, I just wanted to re-emphasize the way I got into this field over the last decade. And I'll make this brief. But I was really kind of atypical. I ended up, atypical for ending up as a career curator. But I think it really speaks to museums and the inspiration and the possibility to explore your interests. And so I started working at the Natural History Museum of Utah about 12 years ago in community outreach. This is me in the window at an event with a bunch of our volunteers at the museum and other museum staff. I was first an assistant and then ran a statewide outreach program, traveling throughout Utah to teach and inspire interest in our natural world. And I did this for about five years. And during this time, I became involved in and helped start the community outreach program at the museum, the Natural History Museum of Utah, which became part of the community outreach program eventually. The focus of this work was to collect data for the city of Salt Lake City to document species and recently restored parks in the city. I would recruit volunteers to help with these projects and hold events called BioBlitzes. But then I was introduced to the collections and sort of the rest is history. And this was still in Utah. I fell in love with this work. I actually volunteered in the collections while working in community outreach and also did field work with the mammalogists. And I was so intrigued by this work that I eventually applied for graduate school to do research and work in the collections at the University of Nevada, Reno. And this is me in the collections in Reno. I came into the program with previous museum experience and so spent the majority of my time in grad school working in this collection. And of course, learning how to conduct research here at the University of Nevada, Reno. And when I came out of this program at UNR, absolute dream job popped up, which is where I am now in museum curation. And this is something that I wanted even before I attended graduate school. And so I'm absolutely ecstatic to have landed this curator position here at such a fantastic place in Santa Barbara. And I really owe all of this to museums. As I mentioned in the beginning, they gave me a place to work hard, and really explore my passions and meet wonderful people who supported me along the way. And I have this purple arrow here because I will absolutely continue to partake in other aspects of the museum work, like community outreach and community science, because I still very much have a passion for this. And with that, I want to thank you and take any questions.
Thank you so much, Allie. We have some questions that have come in the Q&A section, so I'm just
Okay.
going to go ahead and read some of those to you.
Sure.
Paul is wondering, are there other mammals who build stick twig midden-like structures beneath conifers in Southwest Utah?
Oh, that's a really good question. Um, Beneath conifers, yeah. Um, off the top of my head, and nothing is prominently coming up. And if the question is because you've seen one, it's probably a wood rat. And that they actually do that. They will use the base of a juniper or some sort of conifer to build their nests as well. Um, it's just much more common to see them within rocks.
Awesome, thank you. And Larry is wondering, is it possible to identify the species from the appearance of the midden?
Ooh, if you get good enough. Hi, Larry. By the time, well, if you know what exists there, and they have various size differences, then yes, I can almost always guess a scenario from a lepida if I know they both exist there. Um, because of one, as I mentioned, their fecal pellet size, it becomes very familiar. But two, just like knowing where they tend to build and Scenaria almost, or the bushy-tailed almost always needs a very big space with like deep within a crack. So they pick the best spots. So yes, if you get good enough, if you're out there long enough.
Awesome. And Lewis is wondering if you could talk about the different chambers within a midden, like we saw in that drawing earlier in the talk, and talk about
Mm.
what goes on inside of each of those chambers.
Yeah, let's see if I can pull that slide up easily. And so what's interesting, and I'm probably not the best person to ask because I have not excavated any of these, but where is that slide? I can find it. Um, there's not, I think, this is there could be more diagrams out there, but this is from 1968. These were actually drawn. From wood rat nests, likely in more like wooded areas, like the two you see up in the upper left. And so, as far as the ones in the rock go, I mean, I think it's a lot harder to figure out what they actually look like and to diagram them because you're not going to be able to completely excavate them. Um, but yeah, as far as the chambers, I mean, they're listed down here. So they do have things like food caches. And I know it says large interior chamber, places where they just kind of escape the heat during the day and or sleep. And they have nesting chambers specifically for that. I assume nesting chambers also might refer to places where they're rearing young, which might be different than where they're resting, depending. And then, yeah, outer passages and random things. So these wood rats, they're actually like a lot of small mammals. They're very particular about where things go. Like they'll have spots, and you can see this outside of the nest too. They have these latrines where they're pooping, and they're very specific about going back to that same place to either mark a territory or poop, or cache. They have particular caching sites, and the same goes in these nests too. And a lot of rodents are like this. They're not messy. They're clean and they keep things orderly.
Awesome. Liz is wondering, what is the most interesting thing you've found in a wood rat midden?
Ooh. I think like bigger bones, like jaws of like cattle. There's a lot of, you can find skulls of cattle and stuff out in the desert if you're walking around because of the long ranching history. And the fact that a wood rat, even a large wood rat that can carry one of those massive, jaw bones back to their nest has always perplexed me. I have no idea how they do it, and they're solitary, so they're certainly likely doing it on their own. Maybe they're not carrying it far, but they are putting it very distinctly on their nest. So yeah, that's always been kind of interesting.
It is. Leslie is wondering, what is it about their urine and feces that forms the black coating we see on the outside of their nests?
Yeah, I don't know the black coating. That's a really good question. I mean, it's just kind of a hardened glaze. There's a paper, a kind of a older paper, maybe from the 70s or so, that describes um, the like elemental content of their urine. In fact, in some of these places, you'll actually see they not just pee on their nest, but they pee. So if you see this rock, for example, here, there will be like white marks all over these sort of prominent places where the rock juts out. And they're like posts that these animals pee on to mark their territory. And they have high calcium content in their urine. And when it rains, that's the only thing that doesn't wash away. And so you actually see these peeing posts remain, and that's the white substance that's left behind on these places where they exist in the desert. But as far as the glaze goes, I'm not sure. I don't know. I don't know what makes it shiny.
Great. Kelsey is wondering, did you notice whether desert or bushy-tailed wood rats were more likely to use caves for their nests rather than rock outcrops?
Hmm. Good question, Kelsey. Hi. I think that it's really opportunistic and it's depending on the wood rat. So again, like that shelter site, let's see if I can get my mouse work. That shelter site that I showed earlier that was highly thermoregulating. Um, this site was occupied by a larger bushy-tailed wood rat. And the desert wood rats are there and at the site, but they'll pick, the smaller boulders. In fact, I have a picture, and this isn't at the same site, but it's kind of surprising. Maybe I can't find it, but it's kind of surprising how small they'll actually the size of the boulder that they'll actually crawl under, because it doesn't seem like it would fit them. They don't really burrow, so it's sort of like where they can kind of squeeze into. I'm not finding it at the moment. But yeah, like the smaller wood rats and the smaller species, but also the younger, smaller wood rats will just find the sites that are left over. And so these are the best sites. These are the sort of, like I said, like the throne of the site, of the entire site. But they'll go anywhere and they'll nest where they can and they'll move around when they become bigger and older.
Thank you. And just so folks know, we have a lot of questions coming in, so we'll do our best to go a little bit long and address some of them, but we may not be able to hit every single question we've gotten this evening. I'm trying to kind of find some themes, so hopefully you feel like your question is represented.
And I'm sorry, Claire, one quick note on that. Here's my email for anybody whose question doesn't get answered and you really want to ask it. Please feel free.
Awesome. Thank you, Allie. Lindsay is wondering if the two species ever hybridize where they co-occur.
Not these two species. That's a really good question. In fact, there's a lot of research on hybridizing wood rat species throughout the American West. Some really great stuff from my advisor, Dr. Marjorie Matoke. at the University of Nevada, Reno. But these two species are too far speciated. They cannot they can no longer reproduce.
Awesome. And Wes is wondering if there are any particular diseases you worry about digging up in middens, either in old or occupied middens, or if there's any elevated concern in any confined space you enter, such as an old cabin or an old mine.
Mm, yeah, smart, good question. um, so hantavirus, I actually don't think these guys carry it, but I could be wrong. They though they do live in places where deer mice carry hanta, and this is a virus that's really terrible to get. You don't want it, and often you catch it in these places like old cabins where it has the pellets of a deer mouse have become really dry and flaky, and you can actually breathe them in. And so when we go into these places, and these are places where wood rats exist, so when we go into these places, we're really careful to wear respirators or at least masks and get in and out quickly if we're doing any trapping in there. Other than that, I'm not worried too much about the plague. They don't carry. I don't think they carry the plague either. But we do handle these guys, and when we do, we're wearing big heavy gloves so they're not biting us or biting through it. They do carry a lot of parasites. That's another thing we are careful about when we're handling them is that we don't have any ticks or anything that have jumped to us afterwards. There's a little concern, but if you're careful enough, you can avoid it all.
Awesome. And Hillary is wondering if there is any like sexual selection component to why some of these objects are brought in. Is that any motivator to why some of these objects are brought into nests?
Yeah, good question. I would think so. I think that there's certainly an attractant for other animals, for other members of the opposite sex. But I think both males and females have this behavior. And so, yeah, within all the stories and wood rat literature that I've come across, I haven't found anything that's really kind of looked at that too closely. But I would assume that it is about mate attraction and maybe showing off to other members of the not just of the opposite sex, but of their own sex. So, yeah.
Awesome. I'm going to do about two more questions and then we'll wrap up just for the sake of time. Larry is wondering what would be the minimum distance between nests of the same or different species?
Gosh, sometimes I caught a few, and I don't know if they were living in the same nest, but I caught a few of two different species at the same trap site. This happened probably maybe 3 or 4 different times across those 3 years. So very rarely, but I think they're really close to one another. And I would sometimes, we'd place traps right at a nest, so we knew that typically we'd assume that that's the nest of the individual that we caught. And yeah, they would be right next to one another, like I don't know, a few meters at most. So they're living quite close together at some of these sites.
Really interesting. And then we'll end with kind of a fun one. Liz Woolsey is wondering if you'd rather be a desert wood rat or a bushy-tailed wood rat.
Thanks, Liz. Oh man, that's a hard one. It's the hardest question I got asked. I love the bushy-tailed wood rats because they're so beautiful, but I think the desert wood rats take the cake. They're just absolute survivalists, and I respect that.
Perfect. Well,
So,
thank you so much, Allie. Before I pass it back over to Greg, I just want to say I'm sorry we weren't able to hit all the questions tonight. But please do email Dr. Coconis if you have a question that you're really curious about that we didn't get to. And now I'll just pass it back to Greg to close this.
Thanks, Claire. Thanks, Allie. Hey, I want to hand it to everybody on the call or in the Zoom room that I think we had over 230 people participating in tonight's chat, which is a record. So I just want to say thank you very much, and I really appreciate the belief in studying small things and learning from them. So, Allie, thank you very much.
thank you all.
Yeah, it
It
was
was a pleasure.
quite interesting to hear about these little guys and how they reveal so much about our environment. With that said, I would like to encourage you all to join us on April 21st at 6 p.m. for our Water Warriors with writer Michael Branch. And again, if you have any questions about the foundation or would like to know more about how we're supporting Great Basin National Park, feel free to check out greatbasinfoundation.org. And with that said, I hope you all have a great evening and go check your middens.
Thank you. Bye-bye.
Bye bye.